ladies and gentlemen. 7 years ago, I was sitting in this auditorium listening to lectures, and I got inspired to choose a career in science. Today, I want to share that excitement with you and give you some insights into molecular biology and cancer. The human DNA is basically the blueprint of our body. It consists of 46 chromosomes, or pieces of DNA, and 6.4 billion base pairs. Interestingly, we have only 21,000 protein-coding genes, and only 2% of the total DNA amount are exons that code the amino acid sequence of our proteins. The rest are regulating elements, introns, genetic junk, and dark matter. However, 99.9% of our DNA is similar with all and every human being on this planet. Mutations are changes in this DNA sequence. They can occur spontaneously during DNA replication, or they can be caused by chemicals or radiation. Anyway, they can cause 3 major things. Most often, they do nothing at all. But sometimes they can change the protein's amino acid sequence and therefore their functions. Or they can change the protein's expression. That means that how many copies are made from the protein's gene. Therefore, the amount of the protein changes within the cell. If these 2 types of mutations happen in the germline cells that create our sperm and egg cells, and these mutations are combined with natural selection, they are the engines of evolution. However, most mutations occur in our somatic cells that form most of our body, and they can only cause diseases. Most important of them is cancer. For example, if a mutation happens in genes that regulate the cell cycle or apoptosis, those cells will divide non-stop without the ability to undergo programmed cell death when they should. If these cells gain other mutations that affect invasion and metastasis, then they can even spread in your body. But at least we can detect these mutations and understand cancer as a disease better, and sometimes we can even target these mutations directly. For example, in melanoma, which is a type of skin cancer, the V600E mutation of the BRAF gene can be targeted with vemurafenib, a specific inhibitor. Or in breast cancer, HER2 genes amplification, which is also a type of mutation, can be targeted with a specific monoclonal antibody. And luckily, this list is longer and it's growing. But besides therapy, detecting these mutations is also very important in diagnosis of cancer. So how can we detect these mutations? Here are some techniques that we can use. I could talk about these until the cows come home, but I would rather concentrate on the first one, PCR. But what the hell is PCR that everyone is talking about? It's a polymerase chain reaction. Let's break it down. Polymerase is the enzyme which copies the DNA of a cell before it can divide. Or it can also copy a DNA outside of a cell in a test tube. A chain reaction is when one event causes a series of repeating events, like a domino effect or positive feedback loop, and we could describe it mathematically as an exponential function. If we combine these two, polymerase and chain reaction, we get a Nobel Prize-winning invention which can diagnose COVID, which can identify murderer, or which can detect cancer-related mutations. Please allow me to introduce you how you can become a wizard of PCR, how you can concoct your very own PCR potion. First, you put very, very pure water in a test tube. You add some DNTPs, or deoxynucleotide triphosphates, that are the Lego pieces that build into DNA. You add some primers that are short, specific DNA sequences for a region, and they serve as starting points for the DNA polymerase, which of course you also have to add. And you add the DNA template that you want to amplify or copy several times. After you have these in the potion, you have to cook it. You heat the mixture until the DNA strands divide. Then you cool the mixture until the primers bind to their complementary DNA sequences. Then you heat the mixture again until the DNA polymerase does its job and elongates this strand and creates a new strand of DNA. Then you repeat it. Heat, cool, heat, and with each of every cycle of this heating, cooling, heating, your number of DNA pieces in the sample doubles. In the end, you get billions of copies of this short DNA strand that are in between your two primers. For example, if you want to test COVID-19 with it, then you have to take a sample from a patient, usually from their nose. You have to isolate RNA from this sample, since COVID is an RNA virus, and you have to reverse transcribe this RNA into DNA, and in the end, you can make the PCR potion with the primers that are specific for this COVID cDNA, and put it into a real-time qPCR machine, which follows, as it was here told by Shahrokh, that it can be followed during the whole process how many double-stranded DNA pieces are in the mixture. And if it reaches a threshold, you know that there was COVID in the sample. You can also use PCR to identify a murderer. For this, you have to collect cells, blood, skin, hair of the murderer from the crime scene. You also have to isolate DNA from it, and then you can make a PCR for the 0.1% of the human DNA that is not similar amongst all human beings. For example, targeting the short tandem repeat parts of the DNA, and that is going to give you a DNA fingerprint. Just like on this picture, in the first column is the murderer's DNA fingerprint, and as you can observe, it's similar to suspect number 3's. They have the 3 bands at the same level. They are the same person, or at least they are identical twins, since identical twins share the same genome and therefore the same DNA fingerprint. So going back to the sad part, PCR can be used for cancer-related mutation detection. On this chart, you can see how important this topic is, since only in Hungary there are 60,000 new cancer cases each and every year, and almost 30,000 deaths. Timely diagnosis and treatment could save a lot of lives. Therefore, in our laboratory, we are working on a method that could detect 2 very frequent key mutations in bladder cancer patients' urine. And although bladder cancer is just 5% of all cancer cases, it means 3,000 people in Hungary each year and 600,000 worldwide. So we worked out a method that utilizes PCR and other magics of biochemistry. For this, we isolate DNA from the urine samples of the bladder cancer patients and made a PCR, after which came a cleanup step and a specific cleavage of the mutant DNA with a specific enzyme called an endonuclease. And after that, we perform a second PCR, which amplifies only the cleaved mutant DNA. In these pictures, you can see some of our results. Here, 14 bladder cancer patients were tested with our method. Their urine samples and their tumor tissue samples, and they gave the similar results. 12 from the 14 patients had mutations in their samples. 2 patients, patient 3 and patient 14, had mutation 2. 2 patients had no mutations at all, patient 11 and patient 13, and the rest of the patient had mutation 1. We hope that sooner or later our method could provide a screening opportunity by which the patients could be diagnosed earlier and therefore treated earlier. That could save some of them. I'm grateful for the support of the foundation and people listed on this slide, especially Professor Boros Imre Miklós and Dr. Vedek Balázs, with whom we worked together on this bladder cancer project. Ladies and gentlemen, I hope that you become interested in scientific research during this conference as I did 7 years ago. Thank you for your attention.
Thank you for the presentation, and now please take your certificate. Ákos Kovács.